Hi, let's go down to the details, the cellular details of the muscle contraction. So when we look at muscle contraction, it is quite complicated with a lot of parts. So this is in the textbook, and it talks about, here is the muscle fiber. Now imagine you're eating a pulled pork sandwich, and each little stringy bit, that's actually a muscle fiber. So if you've ever made pulled pork, you take a nice big slab of beef tenderloin, and pork tenderloin, and you cook it slowly with heat and a little bit of acid. And slowly the acid will break down the connective tissue, and you end up with just the stringy fibers. Now, there's a lot of terms that go with those, and each one of those stringy fibers is actually a muscle fiber, and each muscle fiber must be innervated by a nerve, and we will talk about how the nerve stimulates it later on. So the nerve stimulates the muscle fiber. It's something called a neuromuscular junction that we will talk about later on, as I said. But what actually causes the muscles to contract is something called the sliding filament theory. So unfortunately, this diagram jumps from here to here without connecting in between. But what happens here is you have in each muscle fiber, there is an area called a sarcolemma, and a sarcolemma is Z-line to Z-line, and that's what causes the striations on the muscle. Now, there are two main proteins that are involved in this. The actin, which is the green stuff, and it's the thin fiber, and then there's the myosin, which is sort of the free floating one, and it's got all the little heads that grab. So when the muscle is relaxed, these Z lines are farther apart, and when it goes to contract, the little heads grab on and will slide the green thing past the purple thing. So it slides the actin past the myosin, and that is why it is called the sliding filament theory. Here's a video that looks at the muscle contraction in a little more detail. So you've got the actin, and the actin is covered, the actin binding sites are covered with this molecule called troponin. So again, too many terms, but realize that the area where, a binding site is sort of like a hand. If the hand is full of something else, say my phone. So if the hand is full, the other part can't bind. So what needs to happen is calcium comes, removes what was in the binding site, and so the actin binding site can then have the myosin head come and bind with it. So a little video to help explain this.
Striated muscle is made up of bundles of multinucleated muscle cells, called myofibers. A myofiber is packed with myofibrils that extend the length of the cell. A sarcomere, the functional instructional unit of muscle, consists of thick myosin filaments and thin actin filaments. Titin keeps thick filaments centered during contraction. In muscle contraction, actin filaments slide past myosin filaments. Neither filaments change in length, yet the sarcomere becomes shorter. The force required for this movement is generated by myosin heads, which make cross-bridge attachments to actin.
Okay, let's stop right now here. So here is the actin, and the actin has the binding heads, the binding sites full. And this is the myosin with the heads on them. Now, when it is full, when the binding site is full, the myosin cannot join with the actin to form what's called a cross bridge.
Myosin heads walk along an actin filament, using ATP for energy. Tropomyosin is part of the complex that prevents myosin from sliding along the thin filaments. A myosin head binds ATP, and as a result, detaches from the actin filament. It rebinds in a new position. Phosphate release triggers a power stroke that moves the actin filament. ADP disassociates, and the process is repeated.
Okay, so you see that there's ATP there, and the ATP is the form of energy. So what happens is energy must be expended in order to unrelease the myosin, and then the myosin will bend, grab, and slide. So it's kind of like a grab and pull, grab and pull, grab and pull, and it will slide one past the other. Good, and now we watch it slowly with no sound. There we go. Okay, so that gives you the idea of what's going on. So when we go to our note sheet, the note sheet is talking about muscle contractions. So we are looking at the muscle contraction. So up here we have our actin, which is our thin, and our myosin, which is the thick one with the head. Then you have how the smooth muscle contracts is the myosin head will grab onto the actin binding site and basically pull. Okay? And that describes it. Now in your textbook, before going on to the other material, it talks about so we're down here, how a muscle contracts. And it goes into a lot of energy. And instead of me walking through, what I want you to do is look at this and then look at the videos that are linked and go through each one in detail. Now, I want you to look at the activity, or so the videos for the animation, but don't get all hooked up on all the details of the naming, because there's a lot of naming. Pay attention to actin, myosin, cross bridges is another one, and then the sliding theory. Look a little too about how ATP works. So go through those questions. And then just one more thing I want to talk about before I let you look at those is the different types of energy. The muscle has energy, and ATP is the most useful type of energy when there's oxygen to break down glucose. Creatine phosphate is another type of energy that is stored in the muscles. Good, so here is a diagram. This is figure 12.4.5. So here you have ATP, which is the major energy source. ATP will bind with creatine to form creatine phosphate. And creatine phosphate is a very quick way to store energy, and it's in the muscle. So if you remember, ATP is made when glucose comes together in the mitochondria, and it produces ATP. Now, there is something called anaerobic respiration. And anaerobic respiration is when there is no oxygen. So if there is no oxygen, the glucose molecule doesn't break down completely. It will only break down partially and result in lactic acid. And a little lactic acid is okay, but too much lactic acid will build up in the blood, changing the pH, which causes a problem. So when we look back at our three types of energy, you have ATP, which is the energy produced from oxygen in the mitochondria to break down the glucose to as much ATP as possible. Creatine phosphate is when ATP, the phosphate group, is stored on creatine as a short-term temporary muscle storage. And then anaerobic respiration is when you get lactic acid, and you do get 2 ATP out of it. So it's not a lot, but you do get some. Okay, so that's the energy. Now, I do realize at the beginning we missed isometric and isotonic. Iso means the same. So isometric means you keep the same length. So that's when your muscle contraction is changing tone. For example, when you're pushing and there's no movement. So a beautiful example is as you're sitting here, your muscles in your spine have tone to keep you upright and give you core strength, but they are not moving. So that is an example of an isometric contraction. In isotonic, you keep the same tone, but you're changing your measurement. And a perfect example of that is the bicep curl. Okay, that is the introduction for this. Go through the videos to look at muscle contractions. And they do a very good job of it. Muscle contraction, and then also cross bridges, and then something called the what's the word I want? Oh, neuromuscular junction. We'll talk about later. Okay, good.